So for German standards, we have waited 5 minutes. I guess we've exhausted our patience. And, welcome, everyone. I'm so happy to see that this lecture hall is filling itself. But I do know that in the live stream out there, there are many more people who are eager to listen to the lecture. So welcome to all of you in the live stream, and welcome to you here in the hall. But most of all, of course, welcome our guest of honor, dear Professor Becker, dear Catherina, dear colleagues and members of the TUD community. Third time is a charm, that's what they say. So believe it or not, this is the third inaugural university-wide lecture, and I think it will be a charm. And you have to listen to it again every third Thursday in the month during the semester at 4:40. This is what's going to happen, an inaugural university-wide lecture. And why in the world did we think in the rectorate, does this university need yet another format? Well, we felt that we have too few formats that actually brings together our 17 faculties, hopefully, eventually, sometime. And that is what this is for. It is to create a space for the TUD Universitas, which is 17 faculties and 5 schools, as you know. And we have 4 campi in Dresden and a little bit outside of Dresden. So we need to find formats that hopefully bring our community closer together. And I think it's attractive. Why would you come here? Because it's presenting strategically relevant professorships and, of course, the unique and outstanding scholars that we were able to recruit for these professorships to our university. And by providing a platform for them to present themselves to our TUD Universitas, we are at the same time introducing the hosting institutions and strategic ideas that are underway in those units, scientific units, teaching units of our university. You cannot catch up in such a large place as we are. 40,000 people is our Universitas, so we have to find ways to keep each other updated. And so I'm very grateful that you came here today and contribute to creating this interdisciplinary space. Because, of course, it is in the heart of each research university, let alone excellence university, to attract the best minds to our university. But not only attract them, but actually welcome them, embrace them, make them a part of our Universitas. And so we have to come together, create meeting spaces, networking spaces to do so. And today, I'm very, very pleased, honored, and proud that we succeeded to attract one of the world-class neuroscientists, Professor Catherina Becker, to our TU Dresden. And not only has she been, as we attracted her, a world-class neuroscientist, but also she brings with her one of the most prestigious science awards in Germany, which is the Alexander von Humboldt Professorship 2022. And it's with 5 million euros, the largest science award in Germany. So it's a double whopper. I mean, thank you very much and welcome very much, Catherina. So let me tell you a little bit. I'm not the one who is giving the extensive laudatio for Catherina Becker, but let me tell you a little bit about Catharina's life before TU Dresden. There has been a life before TU Dresden. So she trained at the University of Bremen, so she's a northern light, as they say, and I'm very fond of the little accent that she still carries, bringing the coast to Dresden, so that's very nice. And then you held positions at the University of Hamburg, at the ETH in Zurich, and the University of California, Irvine. before she got lured back to Europe. At that time it was still called Europe. It's the UK, and more particularly Scotland, the University of Edinburgh. And I think it's fair to say that Catherina Becker was one of the main contributors to make the University of Edinburgh move up the ranks during her tenure in the ranking that the UK has been exercising for their research universities. So we have great hopes. Catherina Becker is considered one of the pioneers in spinal cord regeneration research, and her favorite animal model is the zebrafish. We will hear about it. So the regeneration of the central nervous system, the brain, and the spinal cord is the focus of Catharina's research. And no one better than herself is able to explain it, and that's what she will do later. I have no doubts in my mind. However, what's also very special, I think, about her approach to neuroscience and her approach to regeneration research is that she is an ideal scholar to connect the life sciences across our Johannstadt campus. And to just give you an example, it didn't take a week, let me exaggerate a little bit, for Catharina to identify O'Shea Compass, who we heard just a little while back, and they spun out a new great idea for O'Shea to actually measure the physics of regenerative processes. So there you go. This is interdisciplinarity, this is connectivity, this is creating new ideas, and I can tell you, we will have a little reception afterwards with a little bit of sparkling wine, which will make interdisciplinary talk even easier. So please take the time and stay with us, even if it's only for 15 minutes, it's worth it. But I couldn't speak about Catherina Becker without speaking about all the great people who she brought with her. And she has already attracted to the TU Dresden. And so, first of all, of course, we are very honored and pleased that we have attracted Professor Thomas Becker to our TU Dresden. He is sitting there in the middle, hiding away, but he cannot hide. Welcome, Thomas, and welcome the postdoctoral fellow, Dr. Anna Maria Opri-Soreanu. Did I say this correctly? Yeah? Is she here? She's not here. Well, she will maybe listen online. So anyway, the two of them you brought from Edinburgh, and we are very grateful. And since then, you have been building a lab. So they're sitting all there. So welcome to all of you as well. So Catharina had been associated with TU Dresden. There is a record that there has been an active recruitment going on for quite a while. So she was linked with the CRTD for many years. And since 2017, actually, she chaired the scientific advisory board. And I saw Michael Brandt over there. So I think you were a matchmaker in forging this alliance. And of course, you all know this, but let me repeat it for those of you who may not know it, that the CRTD is one of the pioneering excellence cluster in Eastern Germany. For more than 16 years, it has conducted excellence research, and it was the first East German cluster of excellence. So it focuses as a unit on discovering the principles of cell and tissue regeneration and leveraging this for recognition, treatment, as well as reversal of diseases. And so there you see the importance of having a life science campus next to a medical school and a university hospital where you can really aim for this very challenging hiatus, from cell to bedside, which is the big challenge of health sciences. And so I'm very pleased that the current director, Federico Caligari, will a little bit later on give you a more extensive introduction, both to Catharina as well as to the CRTD. And Michael Schröder, who is the current director of the CMCB, the Center for Molecular and Cellular Bioengineering, under which a CRTD, B-Cube, and biotech are forming a very unique and strategically forged interdisciplinary unit in the health sciences, biomedicine, and bioengineering. So Michael Schröder will give you also a little bit of an introduction. So before Michael and then Fede will come to the mic, however, I would like to thank everyone in the audience who has made this lecture possible. And of course, first of all, there's my chief of protocol, Christine Milkau, but then also from the CRTD, Eike Lau, who has been crucial in bringing this together, and our colleagues from the communication office, and our chief communication officer, Marion Schmidt, is in the audience. So thanks, as always, for your support. And now, without much further ado, I turn the mic over to Michael Schröder, and then we will hear from Fede and finally from our guest of honor. Thanks for being here.
Yeah, thank you very much, and welcome, Catharina. It's lovely to see you here. And Ursula, we heard it from you. CMCB, CRTD, biotech, B-Cube. There's also Paul. Over there is the biology faculty. And of course, in our Johannstadt campus, there are much more. There's DZNE, there's PLIT, there's MPI, CBG, CSBD. There's also PKS. It's awfully complicated, isn't it? It's actually, congratulations. You managed to correctly pronounce all of these acronyms. It's difficult for everybody. It's difficult even more to know what these acronyms mean. And I think the most important thing is that these acronyms, they don't really matter. What matters is the people behind the acronyms. And Catharina, you are one of those people, and you are a very important person behind those acronyms. And I just want to say two kind of personal things before I hand over to Fede. And one is I work together with Catharina. She is the deputy CMCB director. I'm the CMCB director. And from the interaction that we two have together, I can only say you are witty, you are very fast, directly responding. Also, in contrast to many colleagues, your emails come often very, very fast. You're a quick thinker, you are passionate, you are strong, very direct. Sometimes that's also tough on the recipient. And you're funny, and that's really, really great, and I really enjoy working with you, and that makes me very happy. And the second thing I want to say is that I believe that it's very important what you're working on. We saw their spinal cord injury. We're all talking endlessly about corona and about cancer and about many, many other diseases. And of course, they're very, very, very important. But there are also other topics, like, for example, spinal cord injury, where there is actually overall less research going on and less money pouring into. And on a personal level, from biotech, I can tell you, we have a former colleague who was suffering from that. And so there's always a personal note also in the basic research that we do, which is, of course, very, very far away from leading to really deep impact in practice, but it's absolutely necessary, and it's the basis for any progress that is made in the clinic. And so, Catharina, thank you very much for being there as a co-worker. Thank you very much for working on the topics you're working on. And with that, I'd like to hand over to Fede to give the detailed laudatio.
Hi, dear rector, dear guests, dear colleagues, dear friends. It's a great pleasure to be here. My name is Federico Caligari. I'm the current director of the CRTD, as we mentioned. Actually, Professor Staudinger has already said 80% what I was planning to. And it's my immense pleasure and great honor to here introduce to you Catharina. She's been already introduced in some aspect here today. And thank you, Michael, to explain the complex interaction, this jigsaw of what we are trying to build. We grew historically as different institutes popping up at different times and for different reasons and with different background and with different people with different focuses. But we are a team, we are together, and exactly as you, Mika, said, is the people who matter the most, having idea, collaboration, interaction. And I think we are going over a very exciting phase, very exciting times, dynamic. I'm here actually in this campus, it seems to me, ever since, long time. I came to Dresden in 2002, 20 years ago exactly. And I came here as a young, junior, inexperienced postdoc coming from a country that's, what is happening here? It was moving, okay. Coming from a country that struggled, where people struggle to do science at high standard due to not very abundant resources. This is Italy. I'm actually originally from Argentina, even worse, you might say. And I have seen Dresden over these times, this 20 years, literally exploding. Amazing. From already, obviously, with a TUD, with a long tradition as a technician university. I've seen the biomedical, biomedicine, biological, biophysics today, campus growing, institute. Max Planck is the one I joined originally, and then we had, I cannot name them all. It was biotech, probably one of the very first. MTZ was built in the meantime. Then there was B-Cube, in the order CRTD, I think. CRTD was founded in 2006 as a Fortune Center of the DFG and Cluster of Excellence, as you know. We have been growing ever since. The Plead, etc. is a fantastic, dynamic, beautiful campus. We should all realize this. We should all appreciate where we are and being part of it and its growth. And as I said, this is not talking about me today, but about you, Catherina. Catherina joined the CRTD roughly 1 year ago, in summer, last year. And from the very start, she has been very active as a colleague, as a friend, in at different levels of all the possible levels that one can imagine. I will mention a few of them. She's a prestigious, famous, world-renowned scientist in spinal cord regeneration. Actually, her story as a scientist, she was attracted to science, she told me, due to her interest as a child, as a young child, to disassemble things and then reassemble them again. I don't know whether you were always 100% successful in the reassembling part of it, but this is actually what leads you to science, this passion to understand how things work inside out. Actually, a passion that at times was refrained, like when her mother prevented her to disassemble the TV set at home, right, is something that naturally led her into science, and particularly so the science that caught her the most: biology, how living matter organizes itself, make tissues, make function, make us think, move, etc., the central nervous system. She was initially interested in studying the optic nerve. We have already an excursus of her CV or her experiences in different institutions, so I can skip this part. And particularly going across different phyla and different organisms like salamander, mouse, and fish. Actually, she has been a pioneer in using the fish as a model organism, and particularly so, as we heard, and we will hear more in a minute about spinal cord regeneration. And this is about the science. She is very active across the full spectrum of what we do beyond, well beyond scientific discoveries per se. As Mika said, in less than a year, she already is part of our CMCB organizational team as deputy director of the CMCB, as well as a member of the Wissenschaftsrat of the CMCB. She has been taking many roles with this, investigating options to build the closer ties with biology, indeed, as we heard. And with that, she brings her passion in promoting what is good about doing science at a good level, at a fair level. She, ever since, also from the time in Edinburgh, she was very active, always in promoting career, being a mentor, a good mentor of young scientists, promoting equality, gender balance, etc., all qualities that are needed in many places, and we hope you to continue. I'm certain you will continue that also in our community, and I'm highly confident, certain you will succeed. So, with that, Catherina, would really like to take this opportunity to once again welcome you in our community. I hope you will enjoy. We will do exciting things together. I hope that you will enjoy Dresden as a beautiful city, a place with a lot of art, and I know that you particularly you have art as a second passion after disassembling TV sets. Thank you, Catherina, for being with us, and I give you the stage to your lecture.
To tear off the microphone on the way down. All right. Thank you very, very much for the very kind introductions of everyone. I'm not so red because of the sun, but because I'm embarrassed about the many good words. Dear Ursula, dear rector, colleagues, family, group members, friends, larger members of the community, anyone out there, dear fellow Humboldtians, members of the Humboldt Club, who so generously welcomed me as soon as I was here. Dear fellow whiskey drinkers, who so generously welcomed me when I was here. Thank you very, very much. I think just after a year, I have roughly arrived, although I'm still struggling with the acronyms, I must say. And just before we set up the computer, I turned the center, or the centre, for regenerative therapies into Center for Regenerative Therapies, because that's how it's spelled. Okay, I'm going to take you through a journey of regeneration, and Federico said regeneration is a passion. And what is regeneration, right? Regeneration is to recreate something that's broken, to repair a function, to repair a structure, and it is an integral part of life. Life is defined by renewal, so that if something non-living does this, it's rather creepy. But living beings in general can do it, and non-living beings usually can't. However, everyone agrees, let's just play this to the end because Christine is phenomenal. There, she's beautiful. There are certain things we cannot repair. And everyone knows humans cannot repair a limb. Once it's lost, it's lost. Humans cannot repair a break to the spinal cord. If you break your spinal cord, then you lose sensory and motor function below the injury. And if you lose neurons due to a stroke, if you lose neurons due to a neurodegenerative disease like amyotrophic lateral sclerosis, then no new neurons are born in our central nervous system. And that is accepted as fact, and that's been accepted as or described for a very, very long time, really long. So this is the also called Edward Smith papyrus that probably goes back 2,600 years before Christ and was written by an Egyptian physician called Imhotep, who was the first person we know of who described spinal cord injuries and also described that he thought these were incurable. Very accurate description from today's perspective. You can read this in this paper. He says it's unlikely to repair, but he also says this is a medical condition I intend to fight with. And that is also something that has been going on for a really long time. So, for example, Galen in the 6th, 7th century dragged pigs into the central square and performed experiments on them to see what happens when you injure the spinal cord. Justus Arnemann here in 1787 writes on experiments on regeneration. And he writes, kaltblütige Tiere, wie Fische, habe ich ganz vernachlässigt. Auch Versuche am Federvieh schienen mir nicht vollkommen anwendbar. Sie können allerdings einigen Wert haben, wenn es darauf ankommt, in manchen Tierarten die Kraft der Regeneration zu erforschen. So he looked at regeneration in various species, mostly dogs, ignored other species, so-called lower vertebrates, fish, and also birds, but thinks these animals could have some value when exploring regeneration in other species. And regeneration after spinal cord injury is a very, very complex process. It needs a lot of study and research. If you think about the process, the spinal cord normally is protected by the vertebral column. It's also protected by a dense meshwork of cells that isolates it from the immune system. And now you break your spine, and there are shards that pierce that outer protective hull. You break over the blood-brain barrier, cells start to die, you get influx of blood, there's an excess of excitotoxicity, dying cells release calcium. And that needs to be repaired, right? You need to close that blood-brain barrier. You don't want bacteria in your spinal cord, so you have to close that off. But at the same time, when you close that wound, you can't generate a scar, a barrier to the reconnection of the connections from the brain to the spinal cord, the so-called axons of the nerve cells. So any debris and any scarring has to be resolved, and usually that goes along with an influx of immune cells that eat up the debris and clean up the general area, so then repair can happen. And this repair consists, as I said, of axons that need to grow over the injury site to make the functional connection again. It's like a digger digging up in the street and breaking water mains or breaking your landline that needs to be repaired so the signal can get to your house again or the water can get to your house again. And also these axons, once they're over the injury site, have to make functional connections and talk to the right neurons on the far side of the injury so everything can work again. And due to the injury, a lot of nerve cells die, a lot of neurons die, and then their function has to be somehow replaced, or the neurons have to be replaced. And apart from repairing the blood-brain barrier and some inflammation, very little of that happens in mammals. So what can we do to tackle that? What we can do is we can look at the pros. We can find species, we can find animals that are similar to us in that they are vertebrates, they have a vertebral column, they have a similar organization to us, and can repair their central nervous system. Now, this movie I've used for a very, very long time. It's down to the enthusiasm of a PhD student in the lab at the time, Bettina Lieberoth, who misappropriated a camera system that was originally used for mice. And what she did is she put a fish into a water tank, flat water, and just watched as it swims. And you're going to see the fish here. And they swim quite a lot. They swim about 12 meters in 5 minutes. And they're always very active. You don't need to tease them or anything. It's an adult zebrafish. Zebrafish are about 2.5 centimeters or an inch in old money from tip of the nose to base of the tail. So small, you can keep many of them in the lab. So they swim quite actively. And we can take those fish and we anesthetize them and we put them on ice and we transect the spinal cord, the entire spinal cord, including vertebral column, so all the axons are transected. And you're going to see the same fish now that you just saw 1 week after such an injury. And the movie is running. The ones in the front row can maybe see that the gills are moving and the fish is going to try and move now. And you can see that this fish has the problem that we would have. There is the injury side, it's sort of mid-thoracic level, and anything behind the injury, the fish has no control over. It can swim upright and can move about with the pectoral fins, and it can feed, and we treat them very well. We feed them off a pipette. So they're surviving very well, but they can't swim, just like I wouldn't be able to walk if I broke my spine. And then we keep them for 5 weeks longer, and this is the same fish that you saw twice already. So the function returns. And that's when we talk about central nervous system regeneration, that's what we're speaking about. We're speaking about the return of this function. We want to be able to walk again. We want to be able to see again. We want to be able to think. And that is what regeneration to us means. Zebrafish are a fantastic model to look at that. But I've just shown you adult zebrafish, and they are about 3 to 6 months old, and they're not translucent, and it takes 6 weeks to look at regeneration. So we're giving away a lot of benefits that working on younger zebrafish, that, for example, O.J. Campos showed you a month ago, has, in that young zebrafish are translucent, and development is very quick. So when an adult zebrafish lays an egg, development goes very quick. And by about 3 days, you have a small fish with a beating heart, with moving muscles, with a peripheral nervous system, with a central nervous system. They can swim about. And because they swim about, they are also liable to predation. Other fish might come and eat them, or other things may come and eat them. So this is such a scenario that was recorded in the lab of Joe Feccio by Dave McLean some years ago. This is a dragonfly larva. These things are always hungry. This is a zebrafish baby. It's 4 days old. And if I play this movie, you see a bit of a nightmare. That dragonfly larva is trying to eat the zebrafish, and the little zebrafish has to escape. So it can do that. So it has a functioning locomotor system. And so we argued a few years ago, if there's a functioning locomotor system, we can give it a lesion, and we can look at its repair, and all of that falls at a time when the fish is small enough to fit under a microscope, and it's translucent, so we can see what happens inside the fish. So this is what we did. We cut the spinal cord physically with a needle. We can also do it with a laser, but we mostly do it like that because it's quicker. Our best postdoc, Daniel Wehner, could do 300 in an hour. And this is a side view of the fish. It's at this level here. And you can see that the injury is quite significant, and it cuts through the entire spinal cord, and it heals over in 48 hours. And I have this little asterisk here to remind me to tell you that at that point, 48 hours past the injury, the fish become 5 days old, and this is when they become protected under European legislation. So we're doing an experiment on a small vertebrate that is moving. We paralyze it, we look at repair, and all of that is not an animal experiment. So it also basically prevents the use of adult animals under the respective legislation. All right. So what happens then? It heals from the outside, that's nice, but does it heal from the inside? Yes, it does. So up here, you have the injury after 1 day. You can see there's a very thin connection, and about 40% of our fish after 1 day have a thin connection like that, a thin bridge of newly grown axons. 2 days post-injury, about 80% of our animals have such a bridge, and that's about where it stays. So about 80% of the fish that we lesion develop such an axonal bridge. That's pretty good and standard. And then if you look at, do they swim better? Here's a cohort that we've not lesioned, and here's a cohort with spinal cord injury. Directly after spinal cord injury, they don't swim. They get a bit better. After 2 days, they're no longer statistically different from their unlesioned clutch mates. And this continues, but if you lesion the spinal cord of both, they can't swim. So we have a system in which we can lesion the spinal cord. We can see loss of function. We can see structural repair to a certain degree. And with that, we can see return of function. So that is a great model system. And all of that happens in 48 hours. So it's super suitable for large N numbers and experiments to look at mechanisms. And that means we can now address questions. And one of the questions that I talked about at the beginning is, how do the axons make it across this hole, right? It's quite a significant gap, and the axons can't sort of jump over that gap. How do they get to the other side? And one hypothesis that has been around for some years, about 10 years now, and that really sticks to the field is that of a glial bridge. So this is from a paper from the Pete Curry group, and they saw that there are these glial cells, these support cells in the central nervous system, and normally they are sort of going from the central canal out to the pia. And after injury, they align themselves basically in the direction that the axons have to take, and you see axons grow in that direction as well. So it's like railway rails that are laid across the injury site, and then the axons come, and that's an incredibly seductive theory, and it looks beautiful. So we can test that, right? And just to show, this is what it looks like in the fish, right? It looks like there's a street laid down. But we can also make movies now, because we have our fish, they're translucent. We can make them transgenic. We can make our cells of interest green or red or whatever color we desire. So on the right-hand side here, you have one fish. And this is the spinal injury site. All neurons are red, all glia. These cells are green. And you can just look at what happens over the site of the injury. This film's a few years old. We can make better movies now, but you can basically watch what comes first. And here you can see an axon that grows to the other side, and you can see that they're glial processes as well, but they're not necessarily growing together. Now, axons and glia like each other, and often you will see behavior where they sort of cuddle and they make it across the injury site together. But if you look at such fish and you count, and how many fish do only axon make it over the injury site, only glia make it over the injury site, or both together, you find that at least in half of the fish, axon make it first. So they don't seem to need those rails. Okay, so we know what it's not, but they have to grow on something. So in the group, we went through a lot of different potential cells that they could grow on. For example, blood vessel cells, or myelinating glia, cells that wrap up axons and electrically insulate them, and none of them seem to provide that substrate. Oh no, wait, I forgot a whole experiment. I'm going to tell you how we know that it's not glia. Because we observed that it doesn't seem to be glia, but we can test it as well, right? We have the power of observation, but we also have the power of biological test. So what we did is we had a colleague who had made a fish, a transgenic fish, using a system that was originally devised in Drosophila, where you can drive the expression of a gene in one particular type of cell. And that gene is a bacterial gene called nitroreductase. And nitroreductase, anyone who knows their antibiotics knows that it is a bacterial enzyme, and you use that, you can give, if you have a bacterial infection, you can give people metronidazole. That is an antibiotic. Nitroreductase takes the metronidazole and makes it into a metabolite that cross-links DNA and kills cells. And so this colleague of ours, Didier Stenier, had made fish in which the promoter for GFAP, which is only expressed in glial cells, drives GAL4, which is a little protein that binds to a UAS sequence, an enhancer, and that then drives this nitroreductase. So if we use this fish, then all glial cells express nitroreductase. Nothing happens. But if we stick the entire fish into metronidazole, then only in glial cells, metronidazole is metabolized into this compound that kills cells, and you get rid of all glia. And that's what you see here. You basically have fish that have been treated with DMSO, and the metronidazole is tagged with mCherry. So you see our GFAP cells here. These are clutchmates of the same fish. They've been treated with metronidazole, and all the glial cells are gone. So we took the fish, we killed all glial cells, we gave them an injury, and we looked what happened. And what happened was, if you look up here, these fish have their glia, and the axons nicely grow associated with the glia. These fish don't have glia, and the axons grow beautifully. So that is the proof then, right? This is the power of the experiment, that it's really not glia. So now I come up to the point where I left off. What is it then? It wasn't glia, it wasn't blood vessels, it wasn't myelinating cells. It needed to be something else. And basically, in Edinburgh at the time, we went through our fish room and went, what else could it be? What else could it be? So there was a fish line in the fish room, originally by Naoki Mochizuki, that labels periventricular pericytes. These are fibroblast-like cells that an unlesioned fish live around the blood vessels. So here they are clinging to blood vessels before an injury. And after an injury, we saw that they accumulate in the injury site. They seem to migrate into the injury site, and the injury site gets strongly fluorescent, and it's there at the time that the axons grow, which was a great prerequisite for being a substrate. But when we looked at the axons, we saw that the axons didn't necessarily grow in association with those cells. But what do fibroblasts do? They secrete extracellular matrix. So in a series of experiments, Daniel Wiener at the time went and had a look at extracellular matrix factors. He actually looked at 47 fibronectins and mainly collagens. And he found that collagen 12A1 was laid down here in green, was laid down in the injury site, and was laid down in fibrils that are orientated in the directions that the axons have to grow. And this is a physical phenomenon that's certainly worth looking at, because the collagen is spanned in the direction of axonal growth. And we saw that the axons were associated with it. And if we removed the collagen 12, then axons no longer grew. If we added more collagen 12s, then the axons grew more quickly. So again, we could experimentally show that this is the substrate for the axons. So here are the axons, and then the picture starts turning in a second. You can see how intimately they are connected to the collagen there. So that shows us that after injury, fibroblast-like cells invade the injury site and express collagen 12A1. They express a lot more ECM factors as well. And that clarifies this problem here. So we have the injury, the axons need to grow across. We know now, after spinal cord injury, axons actively navigate, and they use ECM to make it across the injury site. So that's good. But how do these axons find their targets, and how does the local function of neurons that have died get replaced? So one thing that we find in the spinal cord of fish is that they still possess a type of stem cell that we also know from, for example, developing mammalian brain. It looks like a radial glial cell. It has a ciliated cell body that sits at the ventricle, and then a long process that ends at the pia. And this is retained in all central nervous system areas of animals that can regenerate. So you'll find it in the forebrain of fish, for example, as well. You find it in the midbrain of salamanders that can regenerate neurons. And we sort of put ourselves in the position of this stem cell. So under normal circumstances, this stem cell will see a lot of physiological signals, right? It will see dopamine, GABA, serotonin, acetylcholine, BDNF, lots of signals that are used for the day-to-day processing of information. And it will see developmental signals during development and also after an injury. Sonic hedgehog wins FGF. It will be exposed to those signals. But it will also see new signals, completely new signals after an injury. And those are the signals that emanate from the invading immune cells, because these come into the injury site and they've previously not been in the central nervous system, at least not to a significant degree. And now they come and they release cytokines mainly to talk to each other and to orchestrate the arrival of macrophages, for example, and neutrophils. But these signals can also be detected from those progenitor cells. So we formulated the hypothesis that if a stem cell sits there and is exposed to all those signals, then if we find out what this cell expresses in receptors, and we look at macrophages and we see what kind of signals do the macrophages release, then we should be able to identify pairs of ligand versus receptor that will tell us more about how the immune system directs neurogenesis from these progenitor cells. I can show you the process, because it's really quite amazing how intimately these progenitor cells are exposed to the signals. So on the left-hand side here, you see the progenitor cells in magenta. You can also already see the looming needle, and this fish reports on calcium signals. So if we lesion, you can see the first thing that happens after injury is this calcium wave that goes through the progenitor cell. And then what happens if you have a calcium signal is it stimulates the release of reactive oxygen species, and that attracts neutrophils. And we see the neutrophils on the right-hand side here, not only neutrophils. in a fish that reports for one of those cytokines that I talked about, IL-1 beta. So here's 30 minutes after injury. You see the first positive neutrophil arriving, and I'm going to play you the next 15.5 hours. You can see more neutrophils arriving at the injury site, and then more and more and more, and then the entire tissue gets very angry, and cells that are not even neutrophils, keratinocytes and other skin cells express IL-1 beta and start migrating into the injury site. And again, if you think of it from the perspective of forces and tensions, you see this cell movement that can pull the ends of the spinal cord together. And then after the neutrophils come macrophages. So here you see the macrophages in this beautiful cyan. And you can see how they sort of hang about the ends of the spinal cord here, the cut ends, and some of them eat and take away bits of neuron, but other ones just sit there and possibly secrete signals that influence our progenitor cells. Are these important? Are these signals important at all? We can find that out again by manipulation. So we can do a spinal cord injury in our larval fish and immunosuppress them. And for that, we use dexamethasone in this case, which is a very broad spectrum corticosteroid that depresses the immune system and also inhibits migration of immune cells. And if we do that, and we count the number of new neurons, we see that there are fewer neurons. So inflammation seems to be important for repair. We can do the reverse. We can lesion the fish and mimic extra infection. We do that by LPS, which are basically surface sugars of invading microbes and making the system more inflammatory. And if we do that and count neurons, we get more neurons. So we get more neurons. So if you suppress infection or immune system, you get fewer neurons. You augment the immune system, you get more neurons. So it seems to be important. So Louisa Drake in the lab went ahead and looked at these pairs, these receptor pairs and signaling pairs, by first fax isolating all progenitor cells and sending them through single-cell RNA sequencing, which captures our progenitor cells, but also most of their offspring. And then she analyzed the gene expression only in those progenitor cells. So they are labeled, for example, by FOXJ1, which labels cells with a cilium. So these here are our progenitor cells. And then she looked at which genes are particularly strongly expressed in this population of cells. SOX2 is also a progenitor cell marker, so only here. And when she looks at that, she finds TNF receptors that seem to be particularly and TNF signaling associated genes that seem to be particularly associated with those proliferating cells. So that's good. There's a candidate. Do our macrophages express the ligand? So for that, she fact sorted macrophages and any cell that had expressed MPEG-1 GFP. And again, she finds these clusters and identifies a group of macrophages here. And if she looks at gene expression, she sees that especially this group that we've identified as macrophages express TNF. Now, TNF, in theory, is a pro-inflammatory cytokine. So we wanted to look further at its function. And first of all, you put the suspect at the scene of the crime. So there are TNF antibodies. Here are our progenitor cells. Here are the MPEG cells, the macrophages, and you can see how they plug in that injury site. The speckled pattern here is TNF, and you can see how it is in very close contact with the progenitor cells. So suspect is at the scene of the crime. That's good. So then we wanted to see whether our crude manipulation of inflammation by LPS and suppression by dexamethasone, whether that influences TNF secretion, and it does. So if you have unlesioned animals, you have hardly any expression of TNF in the injury site. If you lesion, this goes up 15 times. If you add LPS, this goes up almost 100 times over control. And if you put the fish into dexamethasone, then the upregulation of TNF is no longer significant. So it has to do with this suppression and augmentation of the immune system, so that's good. So then you can inhibit TNF action pharmacologically Pomalidomide is a relative of thalidomide, which we all know from the Contagan scandal, and it inhibits the release of TNF from cells. And if you treat the fish with pomalidomide and inflict a lesion and then count the number of neurons, you get fewer neurons. So that's a pharmacological inhibition. And then you can use different ways to mutate the TNF gene. The CRISPR technique has really revolutionized what we can do in the fish. We could previously not target genes specifically, but now it's as easy as 1, 2, 3. So we did that, and then 2 different mutations of the TNF, or 2 different mutation paradigms, showed that if you have a genetic mutation of TNF, you also get fewer neurons. So pharmacology shows that TNF is important. Genetics shows TNF is important. But you're doing all of that in the entire animal, so it's not clear that this is direct action on the progenitor cells. In order to do that, we FACS-sorted the progenitor cells and incubated them for 4 hours only in zebrafish or human TNF, and we see that transcription factors associated with neurogenesis go up. Okay, good. We have release of TNF from macrophages, and we have binding to the stem cell, and we have neurons on the other side. What happens in the middle? In the middle, you need a change in gene expression program. You need the cell to switch from, I'm just sitting there not doing much, to I am now all excited and make lots of neurons. And that is usually mediated to changes in chromatin, and these changes in chromatin are mediated through things like HDAC, an enzyme that modifies chromatin. So we looked at HDAC, and we saw indeed that if we treat our zebrafish progenitor cells with zebrafish or human TNF, then HDAC mRNA goes up. And we went back into the entire fish and overexpressed an inhibitory form of HDAC only in our progenitor cells. So this is a cross-section. Only in the larval progenitor cells did we overexpress the HDAC. And if we do that, we get fewer neurons again. So now the chain is complete. So we show injury induces the arrival of the macrophages. The macrophages secrete TNF. The TNF binds to the progenitor cell. This leads to the augmentation of HDAC, and when HDAC is upregulated, you get new neurons. Cool. So now we've clarified everything, right? We've shown how axons grow, and they grow on ECM, and we've shown that TNF makes new neurons, so everything's peachy, right? The nervous system is repaired. And it isn't, at least, we don't see a complete recreation of what was there before. Because when we look into the spinal cord, we see cells that we normally do not see. And these are these BCs here. These cells think they're motor neurons. They should be sitting here like their siblings and project down to the muscle to move the muscle. And instead, they sit at the injury site and they project over it like a bridge. And in mammals, if you try to apply therapy to the mammalian spinal cord, some people put stem cells into the injury site to create such a bridge, and it seems that the fish might be doing this spontaneously. So you lose some neurons, and you get new neurons, new building blocks, new degrees of freedom to wire your central nervous system, and that is exciting also from a sort of systems theory perspective, and something that we definitely want to look at. At the moment, Tom Ing, a PhD student in Edinburgh, is looking at that with lineage tracing, looking only at new cells, and Stephen Enos, postdoc in our lab, is following that up with more sophisticated constructs. Okay, so looking forward, what else are we doing? So we want to look at how the network recreates itself, but we also want to understand what other signals act on our progenitor cells. And this is something that Markus Westphal, postdoc in the lab, takes forward with the Dresden Concept Genome Center, which is one of our fantastic technology platforms, which was one of the major attractors also in Dresden that we have these. So basically, what he's going to do is again to look at the progenitor cells, look at the gene expression much more finely timed than we have so far, but at the same time look at the gene expression, potential gene expression profiles, programs by looking at accessibility of the chromatin, and there are new methods to do that. And that gives you per cell, each individual cell, it gives you its gene expression pattern and it gives you the state of the chromatin. So that is really unrivaled resolution to understand how a stem cell might go from quiescence to a neurogenesis program. Federico Kessel in the lab is looking much more closely at what the immune cells do, because they're coming in, and you can see in green the neutrophils very excited. You can see a bit more sluggish the macrophages, and they might be doing much more than just secreting factors, because, for example, if you look at here, just under the injury site, you see that they extend arms, and they might be applying physical forces as well. And what she's doing at the moment, you're seeing 48 hours of regeneration in 1 minute here, 48 hours of continuous monitoring of regeneration, which is what I've wanted to do for ages and which I can do in Dresden now with the help also of the LMF, another technology platform. She is registering the shape of the macrophages at each individual time point, ultimately to correlate gene expression and shape and ultimately function of those cells. Can we do anything with this? I mean, we're playing with fish, they're pretty. Can we do anything with this? Mammals have very similar progenitor cells to us. This is a cross-section through a mouse spinal cord. It looks like a zebrafish spinal cord. And if you lesion this, these cells start to proliferate. They just do the wrong thing. Instead of making new neurons, they make scar. So we want to find out what to base these guys in so they make new neurons. And one way of doing that is, and I'm always at the end, is this fancy machine here that we also have in the Center for Regenerative Therapies, which allows us in vivo screening of zebrafish here. They live in these little 96-well plates. We have a robot that picks them up from the 96-well plates, transports them to a little glass capillary under the microscope. It automatically turns them and then looks through the fish, scans through the fish. So this allows us the screening of hundreds of drugs in a week. And this is something that Ana Maria Oprijorano is currently doing. And then we are translating, trying to see whether drugs that we find have an effect in mammals as well, with Sam David and Rubén López Valles at McGill and UAB in Barcelona, respectively. Come on, yes. And then finally, we do not only compare to mammals, we also compare within fishes, because if there is a minimum requirement for regeneration, if there is the decisive transcription factor, if we don't go quite as far away as a human, but we may look at species that are much more closely related, we might find this deciding factor. So currently, we have a new collaboration with Dr. Alexander Froschauer here from the biology department, which was mediated by Oliver Zior, who's in the audience here, over a whiskey. And Alberto de Camposera is looking at regeneration in the medaka, which is a much closer relative to the zebrafish as humans are. And we know that medaka have a slightly different immune system from zebrafish, and we know that from heart regeneration. And our first experiments indicate that medaka, although it's a fish, and we know that fish regenerate, it is generally known, medaka seems to, even as a larva, regenerate significantly more poorly than zebrafish do. And then finally, Stephen is the person to work with O.J. Campas and Claudia Fröb in his group. You may remember these little oil droplets that O.J. talked about a month ago. These droplets that show you the tensions and the mechanical forces that are going on in tissues that are moving during development. So what Stephen has just done, this is the first fish with lots of beads or lots of little bubbles, oil bubbles that he's injected, is he's injecting the spinal cord with these oil bubbles. He will then inflict a lesion and see how the tissue tension and mechanics change due to the injury. We would expect it to be there a lot more slack, and then how it changes as the immune cells come in. And again, we can just image that, and that is in collaboration with Paul. Okay, lots of work, finally the end. This is not even everything we do, but it is it's some of the things that the group is excited about, that we've brought here, that we hope to carry on, that we have the infrastructure and the enthusiasm and the new team to carry out here and carry further. I mentioned names across the way. The spinal cord injury work was done by Themis, Daniel, Leonardo, and Luisa. Tom is doing the new cells. So this is a group that is still in Edinburgh. Rachel will join us this summer. Obviously, Thomas, my partner in life and in science. And then we have the new group here, Eike, whom you will have encountered when organizing this meeting, but also is organizing my entire life around the science at TU Dresden. It's really fantastic to have someone who's so supportive. And the new group here in Edinburgh, these people have given fish and are collaborators. These people have previously given money, and we are currently supported by the BBSRC, the very generous startups from TU Dresden, and the Alexander von Humboldt Foundation, and patience from everyone who hangs in there while I'll talk and talk and talk and talk. Thank you very much. That's an excellent question, and we've always, so the question is, do these fibroblast-like cells, do they carry the axon, so to speak, or do the axons really actively grow on the ECM? And everything we know from how axons grow indicates that they will be actively navigating that environment. Axons at their tip, for example, also secrete matrix metalloproteinases and other enzymes that modify the extracellular matrix, so we would expect there to be quite an active interaction between the axons and the extracellular matrix. I would expect that you could, and we could, if we wanted, we could probably let the cells lay down the ECM and kill the cells and leave the ECM, and the axon growth would still work.
No, one first.
Oh.
I believe. Right.
Oh.
1, 2, 3.
Franziska.
Thanks for the nice talk, beautiful talk actually. So I was very intrigued by the 48-hour movie of the neutrophils and macrophages, and I think I saw neutrophil reverse migration.
Yes.
So I guess you see that there are two. Is there a way to keep the neutrophils there longer than needed, and what effects would this have?
Yeah. We've not, so the question was, is there a neutrophil reverse migration? So reverse migration means basically migration out of the injury site after they've migrated in. So we've not inhibited reverse migration, but there is a mutant that we have that has more neutrophils. So therefore, there are more neutrophils that are lingering at the injury site than in control embryos. And in those fish, the regeneration, axonal regeneration, is worse. And if you either reduce the number of neutrophils or you reduce the amount of IL-1 beta that they secrete, then axon regeneration improves. I think, Anna, yeah.
Thank you, very inspiring. I was wondering, most of the factors you have described and most of the players, we have as well. So why can't we do it? What is different in the fish, really, from us?
That is a question that comes up every time, right? What is the difference? And I don't think there is regenerin. So I don't think there is the one factor that will influence regeneration and that acts like a switch and you flick it, or a gene you overexpress it, or a gene and you reduce it and then you will get regeneration. There are lots of factors that are important for successful repair. One is, for example, the avidity with which an axon grows. in mammals' central nervous system, axons have quite a significant degree of maturation, and as adult axons do not like to grow very much. This has to do, for example, with transport of integrins into the axon itself, etc. And you can modulate these factors, and then they grow a little bit better. Fish axons are a lot more like peripheral nerves. They really like to grow. And so intrinsic motivation of the axon is one factor, but it's not all. Then how the axon reacts in contact with ECM. Fish also have inhibitory ECM molecules, and in mammals it's thought that inhibitory ECM, inhibitory extracellular factors make growth cones collapse, and then regeneration doesn't happen. But fish have the same molecules, and they do express them after injury. So for example, the spinal cord of zebrafish is full of chondroitin sulfate proteoglycans, and in mammals, CSPGs are inhibitory to axon growth. So what we think at the moment, and it's not quite as a catchy thing as Regeneron, is that there's a balance of positive and negative factors. And the more positive ECM you express, the easier it is for the axon to grow over that ECM. And the less promoting ECM you express, the more difficult it is for the axons to cross, for example, as one factor. But it's definitely multifactorial, and I would also think that therapeutic approaches ultimately will have to be multifactorial. And I should say, and it is a horrible oversight, that I should have mentioned Anna Pötsch as a collaborator on that sequencing project, because she's an expert in large data and DNA damage, which is similar to DNA and chromatin modifications through HDACs.
Counting so many questions. So you are next, Seth.
So thanks for a great and very far-ranging, wide-ranging talk. Do you know how the collagen fibers get oriented across the side of the lesion?
Wouldn't it be great if so it would be great to tag them as they do that, and I presented this data for the first time at a Gordon Research Conference some years ago, and everyone got really excited about that. There are some labeled tagged collagens, so for example collagen 1, and we've tried collagen 1, but it's under the wrong promoter, and it's sort of everywhere, mainly in the skin, and we can't really see how they assemble. Daniel Wehner, who was the postdoc in our lab who did this study, is now a junior group leader at the MPI in Erlangen, and he is currently trying to tag collagen 12 to watch the fibril assembly. What is definitely clear is that there is tension in the injury site. And you see that by how the tissue moves, and you see it in this movie, how the cells move into the injury site, and these forces will play a role in directing the collagen assembly. And we can't measure them quite yet, but we're at it. So come again next year, we'll be able to tell you a lot more.
Great talk. Thanks, Catherina. You showed in the beginning fibroblasts are important, then in the later part, macrophages are important. They, of course, talk to each other a lot. Did you look in your system how they talk to each other? I mean,
Where's
I
Özge?
think
Özge, see Özge there. Özge is a PhD student in the lab. She started she started this year. And she's going to look at exactly that. At, well, A, how they interact, B, whether they may be the same thing, ultimately, right? How, so that's her PhD project. We're going to look at that.
Yeah, I was. Yeah, I was wondering. You said you don't need the glial cells, right? If you kill them, you still get a bridge. But does the spinal cord regenerate completely as well as in other cells? So you don't need them at all, or do you need them as support cells in some way?
That's an excellent question. So the question was, I said we don't need the glial cells at all, and that's of course a very, very bold statement. And a statement that only holds true for the 48 hours that we look at the larval fish. Now, we tried to recreate this experiment in adult zebrafish, and they really, really don't like it. Glial cells are obviously a lot more than just a rail, a guide rail, or a support cell. They're really, really important for ion homeostasis, homeostasis of excitatory neurotransmitters. They do make contacts. They even respond to action potentials from neurons. They're active participants in maintenance of the central nervous system. And I think the only reason that we could do this experiment in larvae is that they're so young and they don't depend on these functions that much. And we didn't keep them past the 5 days, and I would fully expect them to not survive for very much longer. I know that the adults don't. So yes, of course, these fish need glia. The sort of question, the section we were looking at is, do the axons need the glia to get over the injury site? And we were lucky that in our experiment, we were able to show that they don't need them for this, but I fully expect that they need them for survival in subsequent days.
I was wondering if different regenerative systems or different tissues that regenerate, even between different organisms, is there some common themes or mechanisms that they all share? Can one learn from one regenerating tissue about mechanisms that work in other tissues?
There are some things that are incredibly, so the question is, can we learn from different tissues that regenerate? And are there common themes? And there are definitely common themes. The one very bog standard theme is that calcium release followed by reactive oxygen species followed by neutrophils. This will happen if you scratch your skin. This will happen if you poke your muscle. And for example, this crosstalk between macrophages and regeneration, you also see in muscle. There was a beautiful, beautiful Nature paper a couple of years ago, also from the group of Pete Curry, who showed that macrophages were basically hanging around muscle satellite cells for a while, seemingly doing nothing, but in reality, sort of propping up the satellite cells and activating them so they could replace muscle. So these, the sequence of immune cell invasion and reverse migration or death is common, is needed, and definitely the timing of these processes might be one of the keys to eliciting repair in non-regenerating systems, because, for example, in spinal cord, what we see is we get inflammation, and then we get resolution of inflammation and sort of the pro-regenerative phase, and then everything regenerates and it's peachy. In mammals, what you see is you get a pro-inflammatory phase, and then you get a restorative phase, and then 2 weeks later, suddenly all the pro-inflammatory cytokines come up again, and you get secondary damage, and then any neuron that has dared to come up, because sometimes, for example, after stroke, you get some neurogenesis, any axon that has started to grow out will be confronted with this renewed wave of pro-inflammatory cytokines, and the process of regeneration is going to be stopped. So we can learn about the timing, for example, of pro- and anti-inflammatory cytokines that are in the injury site.
Last two questions. 1, 2.
Yeah, wonderful talk, thank you. So, yeah, I was wondering, so it seems that in the fish, fibroblasts are part of the solution, while in mammals, fibroblasts are part of the problem, right? So if I understood it correctly, in the fish, we have these collagen strands spun by the fibroblasts, which are then the track for the neurons to bridge the lesion, while I think that in mammals, the fibroblasts are generating a fibrotic tissue in the lesion, which is then actually clogging lesion, doesn't let the newly formed axons through. So can you speculate a little bit on why do we get this difference? I mean, why are the fibroblasts making it in a particular aligned way across the lesion in the fish? How do they manage?
So the question again is fibroblasts, they seem to be doing the same thing, and which is secreting ECM, and in the one animal is inhibitory, in the other animal it is promoting repair. I have to be really boring and come back to the balance. There is a nice proteomics study from the lab of Liz Bradbury at King's College from some years ago, where she and her team looked at the ECM composition in the rat spinal cord after injury. And there's nothing particular about that ECM. There's nothing that would distinguish that ECM in presence of proteins from ECM in fish. However, I think we need to look at amounts. We can look at side chains, for example, sulfations. We have Lizzie Lake, a PhD student in Edinburgh, is working on heparan sulfate proteoglycans. So sulfation patterns might be different. So we might be looking at core proteins that look very similar. Sulfation patterns might be slightly different. So there may be a change there. That's something that we are exploring. And also, again, referring to Daniel Wiener, who's doing some proteomics with his PhD student to look deeper into the exact, also quantitative ECM deposition in the fish spinal cord.
Well, thank you, Catherina, for bringing it in a nutshell, all this complexity. Forgive me for a probably very naive question from a developmental psychologist. It got me excited to think of the synergies or asynchrony or dysfunctionalities between the genetics of development and the genetics of repair. And is the genetics of repair or the process of repair actually touching back or reactivating also a developmental logic, or is it a completely different ballgame?
Yeah, that's a very good question. So the question is essentially recapitulation or what is happening in terms of gene expression during repair, is it the same as development or different? And it is not totally different. So there are a lot of genes that are re-expressed, a lot of genes that are reused, cell adhesion molecules, for example, that are needed for the growth and directional growth of axons are switched on again, upregulated. That even happens in mammals. So in my prehistory, I dabbled in mice a little bit. So they do upregulate growth-associated molecules initially, but then that stops after a few weeks, and regeneration doesn't happen anymore. There's a lot of approaches also in terms of rewiring the brain after something has gone wrong. So for example, in myopic individuals who lose their functional eye, to reinstate a plasticity and a plastic period to allow rewiring so that they can look through the other eye. And that again goes through modifications or through a turning back of these maturation processes that I'm talking about, that in part are axonal, so they're intrinsic to the neuron, but also in part are in the environment, in the extracellular matrix, that is laid down fully formed synapses and keeps them in place, the so-called neuronal nets. And if you dissolve them, then there is a new phase of plasticity where you can then do, for example, physiotherapy or occupational therapy for your eyes to get some of the vision back. So some processes are similar. Some processes are not happening spontaneously, but can be induced by removing some of the maturation factors that are laid down and during aging of the human.
Great. Please join me in a great applause.
Thank you, yes.